Bismillahirrahmanirrahim. I'd like to talk about now eukaryotic cell structures. Eukaryotic cell structure, in other words, stuff that you find in eukaryotic cells. Now, cell wall is one such thing that you find. Cell wall is a thick, non-selective polysaccharide structure. Example, cellulose. Now, plants have cell wall. Okay, people don't have cell wall. Cell wall is what gives plants the rigid structure. Okay, obviously plants that are very rigid have more cell walls and more cellulose. You'd be surprised that the cellulose, the plant bark, bark for example is an example of a sugar structure. There are sugar structures, and they don't taste like sugar, obviously, because we can't digest that type of sugar. We don't have any taste buds for that, thankfully. But cell wall is a rigid structure. It's like cement, basically, so to speak, and it's very thick and it's non-selective. It does not have the properties of the plasma membrane to select out certain things and not select out certain things. And it's made of sugars, polysaccharides, meaning long chains of sugars, like very long chains of sugars. Okay, example of this obviously is cellulose, is one type of this. Okay, now the reason we can't digest cellulose is because we don't have enzymes. The human beings don't produce enzymes to digest cellulose. But there are plenty other polysaccharides, sugars that are similar to that structure that we can digest. So as simple as that. If we did have the enzymes to digest cellulose, in fact, we could probably even eat bark, but we can't. Anyways, so cell wall is the outermost structure in a plant cell. Right underneath it is the cell membrane. As you can even see in this cartoon picture, the cell membrane is not nearly as thick as the cell wall is. And inside is contained in it is the various different structures we shall talk about. Now, cell wall is not pure cement. It actually has some degree of complexity to it. Plants communicate with one another. Plant cells communicate with one another. So it's like this. They have little tunnels. Okay, these are called plasmodesmata. They're little tunnels between plant cells, which allow them to communicate with each other like this, like a little window from one cell to another cell, like that. Okay, so plasmodesmata, they allow plants to communicate with the little pores, and they obviously would have to go through the cell wall to be able to do that. So this is the cell membrane, of course. The yellow part, and the green is the cell wall. Cell walls provide rigid support to the plant and protection for the plant from the harsh environment, right? Okay, and there are pores from here to here. Okay, this is a transmission electron micrograph of a cell wall, the thick structure of a cell wall, and you see tiny. This here is the plasma membrane. So comparatively, cell wall is much thicker. So here's another same depiction of the plasmodesmata and the cell wall. So one of the distinguishing features between an animal cell and a plant cell is that animal cells do not have a cell wall. Okay, they both have cell membrane. In fact, all cells have to have cell membrane. Okay, all cells have to have cytoplasm. All cells have to have some kind of genetic material. Okay, whether it's the nucleus or not, that's different. In eukaryotic cells, the genetic material is collected in the nucleus. In prokaryotic cells, it's not. But the key distinguishing feature between an animal cell and a plant cell is the fact that animal cells do not have a cell wall. One of the distinguishing features of a plant cell, on the other hand, is a gigantomatic vacuole, a huge vacuole. Plant cells typically have a vacuole filled with water. That's what gives plants turgidity, the strength. In fact, when the plant is dehydrated, it wilts. So here's a plant, it's wilting, and it's like this. But then if you water it, it becomes all nice and strong like this. Okay, so that's the difference between a plant that's dehydrated and a plant that is it's not dehydrated. And when you give water over here, the water collects in the vacuoles, and that's what gives the plants its turgidity. Okay. So vacuoles are very important. They're not just there to give the plants turgidity. The water can also be used for different purposes, but that's a vacuole. So here's a nucleus. It's a beautiful picture. This one, it has nucleus, okay, and another here is the plasmodesmata, which we talked about, and we talked about the cell wall here. And this is a light microscope picture of a plant cell. As you can see, the plants are cell walls are like this. In fact, if you remember Robert Hooke, who described the word cell, he looked at the shells of a cork tree, right? Of an oak tree, and everything inside was missing, and all that was left there was the cell wall. Okay. Now, cell walls are made of cellulose. Cell walls are made of cellulose. Okay. Now, if you magnify the cell wall, and you take one strand, it's made of millions of threads. It's like a blanket. It's like a blanket of threads, right? And each of those little threads, if you super duper magnify it, it'll go like this, and you will see it's made of sugars. The same sugar, glucose monomer. Okay. Remember, glucose plus fructose give you sucrose, which is table sugar. It's sugar. That's sugar that you eat. But the same glucose that's in the sugar is in cellulose. Okay, so but connected differently. Okay, so the way sugar monomers are connected, the pattern of connection is what determines different between different types of polysaccharides. The pattern of connection determines whether it's going to be cellulose or other types of sugars or starches. Okay. So that's the basic structure of the cell wall. Now we shall talk about the nucleus. Okay. The nucleus of the cell is obviously where the DNA is. It's very, very important. Okay. The nucleus has a membrane of its own. That's called a nuclear envelope. Now, why is it called a nuclear envelope? Why does it not call it nuclear membrane? Well, you can also call it nuclear membrane, but it's more than just membrane, see? It's more than just membrane, and that's why you call it a nuclear envelope. In fact, it's two membranes. It's like one membrane on top of another membrane. You'll see in just a second. So it's like two blankets, okay? And the nucleus has holes in it like this. They're called nuclear pores, and that's obviously important because if things want to get in, or what's the nucleus is good for if things cannot get in or get out, right? So for things have to get in and get out, and they're only coming out through these nuclear pores. Inside the nucleus, you frequently see an area that's darker in color than the rest of the cell, and this area is called the nucleolus. The nucleolus is the place where ribosomes are assembled. Okay, ribosomes are very important little thingies. Ribosomes themselves are made of two units, and it's easy enough to remember. One is called a small unit, the other one's called a large subunit. So ribosomes have subunits, small and large. They look like little, gloves, okay, like mittens. So nucleolus is where the ribosomes are assembled. Now ribosomes, they don't get enough respect, I think, or attention, because they do a lot of work. They make proteins. Man, they should be like famous. They should be like the story of ribosomes and the rest of the nuclear or the rest of the cellular organelles. But no, it's just they don't get enough attention. But ribosomes are very, very, very important because they make proteins. But they themselves are assembled. They're made in this dark area of the nucleus called the nucleolus. The rest of the nucleus is surrounded by DNA material. Now, here's an interesting picture. Now, this is DNA of a cell. This is DNA of the cell over here. This is DNA of a cell. This is DNA. But you see, what the heck? What is this, right? This is also DNA of the cells, okay? But here, the DNA is like in a spaghetti form, but here it's highly organized, like in a hot dog form, okay? So when the DNA is open, is called chromatin. That's like in spaghetti form. When the DNA is all wrapped up into very dense, condensed chromatin form, that's called chromosomes. Okay, so when DNA is condensed, it looks like chromosomes. When it's open, like spaghetti, that's called a chromatin. So you have to understand chromatin and chromosomes, they are basically DNA. One is all opened up, ruffled up, and one is very condensed DNA. So chromosomes are condensed chromatin. And these cells, in fact, are different stages of their lives, and that's why they look different. This cell is about to divide. The only time you see chromosomes, mind you, I'm pausing for effect. The only times you see chromosomes, mind you, is when the cell is about to divide. That's the only time, because this chromosome, the DNA right here is pretty useless. I mean, how are you going to go in there and try to find a gene and it's all condensed or rolled up and packaged up like, like in suitcases. So the only time you see a chromosome in the cell is dividing. So how does the DNA get wrapped up? So, okay, here it is. So here's the open DNA. Okay, it gets coiled up. Okay, it gets coiled up, like, and you take it like little beads, like, like marble beads, and you take it and wrap it around the marble beads like this. And you take a whole bunch of marble beads and you wrap it around like this. And you coil those marble bead thingies over here like this. And then you coil that up over here like this. So there are different levels of coiling, right? There are different levels of coiling. So you could do like this, first coiling little tiny marbles, they're called histones, okay? And then you take those histones and you coil them up a little like this, and you supercoil that and supercoil, and then you get a chromosome. So this is a chromosome. This is a coiled up DNA, okay? Now, the chromosome looks like this, and typically has this one area that's called a centromere, which we'll talk about that in a little bit more detail. So this cell is about to divide, and that's why you see them chromosomes. So here's another demonstration of the same. So this is chromatin, opened up DNA. This is the nuclear pore. Now here's the interesting picture here. As you can see here, the nucleus has two membranes. They're like one, it's like plasma membrane, right? It's like two plasma membranes, one like this, and then there's another bilayer like this, a bilayer. So nucleus has double membrane. That's called a nuclear envelope. So chromatin that's coiled up like this becomes a chromosome. In the middle of this, as we said, is called a centromere. And each of this is called, very brilliantly, is called the arm. This is called the short arm of chromosome, and this is called the long arm of chromosome. Ingenious. I agree. So this is a basic structure of a chromosome. Now, you've heard that one before, right? Chromosome. What is a chromosome, right? So now you know what a chromosome is. Now, what you see over here is human chromosomes. Okay, human chromosomes. Okay. Now, here they're all lined up. They don't line up like this. Okay. They don't, like, okay, come on, guys, family portrait. No, it's not like that. So what happens is they find a cell in the human being, and then that's dividing, that has chromosomes, and they take a picture of it, and they put it in different different colors and shades, okay, and then they paste them, okay, they put them over here, and they put number 1, 2. So that's how you're getting this picture over here. But this is a family portrait of a human chromosome, okay. So this family portrait is given a name. It's called a karyotype. K-A-R-Y-O-T-Y-P. I think karyotype. Let me try to spell it. I think, but we'll double check that in the future, okay? The karyotype. It's like a family portrait of the chromosomes. Now, this is very important. So every chromosome, every chromosome has a pair, okay? We have 21, 22, 23, okay? We have 23 pairs, which means we have 46 chromosomes total. 46 total, 23 pairs. Why are they in pairs? Because this is from mom and dad, and it's like that. Mom, dad, mom, dad, mom, dad. We get one from mom, one from mom, dad, one from mom. I don't know which one. Maybe this is from dad, it's from mom, dad, mom, dad, mom, dad. Now, so we get 43 pairs of chromosomes, one from mom and one from dad. Now, interestingly, what you see over here, if you color code them for particular genes, you see these chromosomes, even though this, let's say this is from dad, and let's say this is from mom, these chromosomes over here, they call it the same because they're talking about the same gene. They may be saying different things. They're talking about the same gene. They may be saying different things. In other words, this dad's chromosome might say, okay, the kid is going to have brown eyes. The mom's chromosome might say the kid is going to have blue eyes, right? So the dad's chromosome here might say he's going to have curly hair. Mom's chromosome might say he's going to have straight hair or something like that. So even though they look similar, they are homologous. In other words, they code for similar things, but they say different things. Okay, they give different commands. So every chromosome is different because they carry hundreds and thousands of genes. Okay, but there are a pair of them, and they're all together. We have 23 pairs of chromosomes. The last pair are called the sex chromosomes because they determine the gender of the person. If a person has two X chromosomes, one X from dad, one X from mom, they are a female. If they have one Y chromosome, they are male. So the last chromosomes are called sex chromosomes. The other ones are called autosomes, which we will also study in the future. So this is very important to understand. This is a portrait of human chromosome. There are 23 pairs and therefore 46 in total, and this is called a human karyotype. The nucleus we talked about. Okay, so nucleolus, the assembly area of the ribosomes. We talked about the nucleolus, site of assembly of ribosome subunits. This is a nuclear envelope. The reason is because it has two actually membranes. It's too cool. The nucleus is too cool to have only one membrane. It has two. Okay. The nuclear pores are important because that's where the RNA and proteins come in and out of the nucleus. Okay. The nucleus has a called cytoplasm of its own. It bathes inside. The DNA bathes inside its own cytoplasm. That's called a nucleoplasm, if you wanted to know. Now, this is a scanning electron micrograph of a nuclear membrane. It's a beautiful scanning electron micrograph of a nuclear membrane, and you can see the various different pores that are over there. These are the ribosomes, which have a large subunit and a small subunit. And the ribosome, which is this, the large one and the small, are the site of protein synthesis. Okay, they make the protein. What do you mean make the proteins? To make proteins, you have to have messenger RNA. Okay, so the DNA that sits in the nucleus is too cool to leave the nucleus. DNA does not leave the nucleus. Okay, it's like a book. It's like a reference book. It's like a reference book in the library. You can't check it out, okay? But you can make photocopies of it. So you copy the DNA, you can photocopy of the DNA. That's called a messenger RNA. So the DNA is like a reference book. You can't check it out at the library, but you can make copies of it. And those copies are called messenger RNA. They leave the cell, and so messenger RNA is like a photocopy of the DNA. And using that copy, okay, and bring amino acids together, proteins are made. Okay, and where this happens at the ribosomes. So ribosomes are protein factories. Okay, now the ribosomes frequently associate themselves with what's called endoplasmic reticulum. We will talk about that in a second. So here's the here's the nucleus of a cell. It's a plasma membrane, and the fluid that's inside, it's like the jelly that bathes everything inside. The fluid that surrounds everything, it's called the cytoplasm. So the fluid outside the nucleus is called the cytoplasm. Okay. So this is what we have talked about so far, and these are the terms that you must need to know. Chromatin is the open DNA. Chromosomes are the wrapped up DNA packaged up chromatin. Nucleolus is where the ribosomes are synthesized. Ribosomes, which have two subunits, large and small, and they make proteins. We appreciate the nuclear membrane and nuclear envelope, and we appreciate also the nuclear pores. Now, I mentioned this. Nucleus is where the DNA is. DNA is a double helix. It's a double-stranded molecule like this, and as I said, it's too cool to leave the nucleus, so it is copied, and the copy is called a messenger RNA, mRNA, messenger RNA. This process is called transcription. Okay, and then the RNA then used as a template, as code to make proteins, which are made of amino acids. Which amino acids come together in which order is determined by the sequence that's over here, and this sequence is determined by the DNA sequence. So the structure of the amino acids based on the sequence of amino acids, that's determined by the messenger RNA, which is determined by the DNA. So that's how DNA ultimately determines the structure of proteins. This process going from DNA to RNA is called transcription, and this process going from RNA to make proteins is called translation. Translation because the language here is different, right? This is the language of amino acids, this is the language of nucleotides. Okay, transcription because it's like a copy, because this language is nucleotides, this language is also nucleotides, because DNA and RNA are made of nucleotides, so that's transcription. Transcription is like dictating. Please transcribe this for me, means like take notes and type this up. Okay, transcription. Please translate this into Arabic, means put it in a different language, right? So transcription just means to copy. Translation means to change the language, and language here is amino acids, and here it's nucleotides. This paradigm, that is, going from DNA to messenger RNA and to proteins, this is the central thesis of biology. In other words, this is the most important theory. It's like the theory of gravity. So as it's equivalent to in physics, this is like the theory of gravity in biology. Okay, this is the central thesis of biology. Okay, this phenomenon. I know it's a lot, but I don't know about you, but I find this stuff fascinating. Now here's the nucleus. Outside of nucleus is this huge thing. It's like kind of like a it's an outgrowth of nuclear envelope. In fact, this membrane here grows into and forms a layers of membranes, stacked up membranes like this, and this is called the endoplasmic reticulum. Endoplasmic. So endoplasmic reticulum is all these layers of membrane over here that just kind of go like this, like a towel, like a stacked up towel. Okay. There are two types of endoplasmic reticulum. The cool thing about endoplasmic reticulum is the abbreviation is ER. That by itself gives it certain notoriety, I think. There are two types of endoplasmic reticulum. Okay, there's one that's the smooth, and then that's the rough. And that's pretty easy enough. That's the smooth endoplasmic reticulum because it looks smooth, and this is the rough endoplasmic reticulum because it looks rough. The reason it looks rough is because ribosomes are attached to those tiny things. Ribosomes, they're the ones that make the proteins. So endoplasmic reticulum that has ribosomes attached to it, that's called rough endoplasmic reticulum. Without it, it's smooth endoplasmic reticulum. Now, why are ribosomes so intimately associated with the rough endoplasmic reticulum? Because many times once they make the protein, they dump it into inside the endoplasmic reticulum, where the protein is further modified and packaged so it can be delivered to wherever it needs to go. So that's the reason why ribosomes are so intimately associated with the endoplasmic reticulum. As we have stated, ribosomes and endoplasmic reticulum together are like the protein factories. Here, the ribosomes. From there, they are modified and packaged, and smooth endoplasmic reticulum helps in that and transported to wherever it needs to go. And you see these are intimately associated and outgrowths of nuclear envelope. It's nucleus, a nuclear envelope going into the endoplasmic reticulum. This is a electron micrograph of rough endoplasmic reticulum. As you can see, this looks rough and it's studded with ribosomes. And over this area, there's a paucity. There's no ribosomes over there, and that's the smooth endoplasmic reticulum. Now, the two are connected, of course, and those are the ribosomes, higher magnifications. And now you see all of these are connected. You don't see some of it over here, but over here you can see that one's connected. This one over here goes over here, so they're all connected. It's like an underground network. And in these, okay, here's the ribosome, as I said. It's making the protein, which is, it delivers inside the endoplasmic reticulum, and it's modified. See over here? It's like this, right? And it's changed to something like this, and modification occurs, and then it's packaged into little vacuoles, and transport vesicles, and it's transported to wherever it needs to go. See? So here it is. It's made by the ribosomes over here and dumped inside, not dumped inside, delivered inside the rough endoplasmic reticulum, where it is modified. See, some sugars added to change the etc., and then it is transported. Here's a cartoon picture of this, okay? Now this is the messenger RNA, the brown thing over here. These are the two subunits, the red and the pink are the subunits of RNA. They're going to come together and they're going to make the protein that they deliver into the endoplasmic reticulum. The brown thing is the messenger RNA. The ribosome subunits come together. They make the protein that enters the endoplasmic reticulum. Same process, and then after that happens, it is packaged. Okay, here's the endoplasmic reticulum packaging the protein. From there, they actually go to this place. This is called the Golgi apparatus, in which further modifies, and then it goes to and delivers it to wherever it eventually needs to go. Okay, so this is like you putting this in the mailbox, okay, and this is like the post office, okay. So you do the thing, you make the letter, you put it in the mailbox, and all the mailboxes are collected and then go to the post office, and then the post office delivers the letter to wherever it needs to go. So this is the Golgi apparatus. It looks very similar to the endoplasmic reticulum because it is. It's also just a stack of membranes. So the smooth endoplasmic reticulum, production, storage of lipids. So as I said, smooth ER does other things besides protein. It's responsible for lipid metabolism. Okay, lipid metabolism, such as the creation of membranes. Where are all those membranes going to come from? They have to come from somewhere, right? So ER looks like this. Golgi apparatus looks like this. The Golgi apparatus, or Golgi bodies, they go by different names. The Golgi apparatus, or Golgi bodies, it modifies, packages, and transports proteins. So proteins, they take a lot of work to make, and between the ER and the Golgi apparatus, and they're eventually delivered. Sometimes they're delivered to the outside of the cell. Sometimes they just need inside the cell to do whatever the cell needs to do. So it's not they always have to leave the cell. They could just hang out in the membrane. There could be a receptor in the membrane. So proteins go from here to various different places, either inside the membrane, inside the cell, or on the cell surface, or outside the cell. Okay, I think this is showing the same thing. Now, so this is secretory vesicle going and secreting. Now, sometimes this picture shows a lysosome. Lysosome degrade things. When the cell takes in something right here, this is an incoming vesicle. It combines with the lysosome, and the lysosome degrades it. It breaks it down. So here's a cartoon depiction of what's happening. So endoplasmic reticulum, it transports stuff to Golgi apparatus, which is eventually makes its way to the plasma membrane. See? Like that. Now this picture also suggests not all things make it to the membrane. Some of them go over here. Yeah. So here's another depiction of what happens. So that's the protein in the RFER. Now it's going to go to the Golgi apparatus. There it is in Golgi apparatus. This is simplistic drawing. Just not showing the various modifications that occur in the protein. So I think you got this. All right. Vacuoles. Vacuoles are storage places. They're like cupboards in the cell. Okay, they're sites of food, enzymes, or waste products. So they have various different functions. They have enzymes. They help break things that the cell needs, or helps degrade things that the cells no longer needs. Okay, they're usually not found in animal cells. Okay, they're more predominant in plant cells. Animal cells don't store as much. Central vacuum, which we talked about already. Okay, the key for this picture is here to highlight the critical differences between a plant cell and an animal cell. Plant cells have chloroplast. Okay, they have a central vacuole and a cell wall. Those are three things that are reasonably unique to plants. Obviously, we don't have chloroplast because we don't have leaves. Okay, vacuoles. Lysosomes are organelles that contain digestive enzymes. They break things up, okay? Either things that are bad for the cell, like bacteria, or worn out organelles. If something's not functioning properly, it gets delivered to the lysosome, it's broken down. Food particles or bacteria or viruses. So the lysosomes are like trash compactors, with digestive enzymes. So anything that comes in, mixes and goes to the lysosome, and then it's done, it's gone. Lysosomes. Now, how do these things move in the cell? Like, how does this go from here? How then this connects? How things move? Things move inside the cell because there's a network of highways like this. Okay, there's a microtubules. There's there are various different webby-like things. They're all scattered about throughout the cell, and proteins attached to, like, for example, this vacuole, and this is a protein. It's like a taxi. It's a mover protein which attaches to this, and attaches to this, and it kind of walks on this microtubule. This is a kinesin motor protein. So one of the cutest things. And it delivers things. That's how things move inside a cell. How do things move? They move by grabbing onto the cytoskeleton, the skeleton of the cells, these little thready fibers that are all over the cell, and they kind of tug along in that way. And this is the kinesin protein. It's kind of cool. It kind of moves things like that. It's a motor protein. And this is just a summary picture, I think. And next time, inshallah, we shall talk about cells energy. Until then, as-salatu was-salamu ala rasulillah. Assalamu alaikum.